What's the structure of a gene? Here's a cell, and I'll draw a big nucleus inside the cell. Within it are many, many genes. Each gene is part of a strand of DNA with these many base pairs. If we zoomed in, we'd have the backbone of sugars and phosphates, and the bases, C's and G's, T's, and A's, pairing up. Each gene may be 1,000 or 3,000 base pairs long. At the beginning of each gene are areas where proteins can bind that can be enhancers, and enhancers make it more likely that that gene will have messenger RNA made from it. Enhancers make it more likely that a molecule called RNA polymerase, I'll just call it RP, will bind. RNA polymerase will start to move along, and as RNA polymerase moves along, it starts making a strand of messenger RNA. When it gets to the end, that strand of messenger RNA is for the whole length of that gene. That strand of messenger RNA may be processed. It may have pieces chopped out of it, and then leaves the final strand from whatever pieces remain. That strand of RNA has G's, A's, U's, and C's. It's a single strand with those bases on it. Each 3 of these is a codon. That codon is going to correspond to a particular amino acid. There are also proteins called repressors, R. Repressors get in the way. This enhancer needs to bind in order for RNA polymerase to bind and start moving. If instead a repressor has stuck to that same spot, to the bases in that location, that may prevent RNA polymerase from binding, and so you won't get messenger RNA from that gene. If there's a change somewhere in that gene, a mutation, a base pair that has been replaced with another, that can happen both in this area where it will affect the structure of the protein, and it can happen where these enhancers and repressors bind. To learn this, one of the best things you can do is practicing sketching for yourself. In the sketch, I'll go through all the things I just talked about and label them as I go, showing you the enhancers and repressors. And when the repressors are there, it blocks transcription. But when the enhancers are there instead of the repressors, that allows RNA polymerase to bind, move along the DNA, make messenger RNA. That messenger RNA gets trimmed a little bit to form the final messenger RNA. If there's a mutation somewhere in the gene, for example, a T and an A base pair change to a C and a G, that will change the messenger RNA that's made. If we've gone from a TA to a CG, here's our changed CG. Matching that G on the messenger RNA would be a C. This is the same strand down below, so here's the C, and it's the same messenger RNA strand, so it's a C. That messenger final RNA strand going out into the cytoplasm would have the mutant, would have a C, where before it would have had a U. That changed base will change the structure of the protein.